Next, specification number 9 says, investigate osmosis using materials such as dialysis tubing. Dialysis tubing is a semi-permeable membrane used in labs to simulate biological membranes. It allows small molecules like water to pass through while blocking larger molecules like proteins and starch. It's commonly used in osmosis and diffusion experiments to study how substances move across cell membranes. Here, we have a diagram showing an osmosis experiment using dialysis tubing. In this setup, water moves from the beaker, where its concentration is higher, into the tubing, where the concentration is lower. This process demonstrates how water crosses a semi-permeable membrane to balance the concentration on both sides. In this experiment, we will investigate the movement of water and the process of osmosis. This experiment illustrates osmosis, where water moves through a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. By using dialysis tubing filled with a sugar solution, we can observe how osmosis influences the movement of water based on the concentration gradient. The procedure involves: start by cutting a 20 cm length of pre-soaked dialysis tubing and tying a knot at one end to seal it. Then, fill the tubing with a concentrated sugar solution mixed with colored dye. Next, attach the open end of the tubing to a capillary tube and secure it with an elastic band to prevent any leaks. Once the setup is complete, clamp the capillary tube so that the dialysis tubing is fully submerged in a beaker of water. Finally, observe the liquid level in the capillary tube over the next 10 to 15 minutes to track the changes resulting from osmosis. The results indicate that the liquid level in the capillary tube rises. This suggests that water is moving from the beaker into the sugar solution through the partially permeable membrane, and this behavior is expected, as osmosis causes water to flow towards the area with a higher solute concentration, demonstrating the fundamental principles of osmotic movement.